Hello, you've just received your envelope from Dr. Ann. So let's unpack it and see what's in it. First is the materials for your protein synthesis lab. We'll do this lab first. You should have a pair of scissors and some tape handy. In the packet, you will have received a white envelope. In the white envelope will be nucleotides, a poly-A tail, and a guanine cap. You also should have received your DNA strand paper, a codon table, your lab packet. This is the lab that we're going to fill in. Also has the instructions in it. And then you received a packet that I gave you with types of mutations. You will also receive a lab for next week. That is the cellular respiration lab. In that, along with your protein synthesis lab, you should have received a piece of poster paper, a packet with the cellular respiration processes that you'll cut out. There is instructions on a second video for this. And then also your cellular respiration practice questions, which will be due for points for the lab. So for this lab, you'll take a picture of the poster once you construct it, and you'll also turn in your lab. So let's get started. For this video, I'm going to teach you how to do the protein synthesis lab. Always the first thing that you should do is go through your packet and read through it so you kind of have an idea of what you're doing and what the instructions are. So read the first 3 pages. It may not make sense at first, but the second time you read it, what you're going to do is go through and do what it's asking you to do. The first thing I did was I cut my DNA strands apart so it was as a sheet of paper. Now it's in linear strands. So the linear strands are numbered. The number is going to go at the start of the line. So where the number is, that's where the engine of the train is. Where the number is not, that's where the caboose is. So let's construct our DNA strand by taping them together. Here's my newly constructed DNA strand. Notice it starts with number 1, it doesn't end with number 1. The second part starts with number 2, it does not end with number 2. So please don't put them backwards. If you put them backwards, you're not going to do this correctly. So here's my DNA strand. All 4 linear pieces of DNA are taped together. Where would this DNA be found in the cell? The next thing you need to do is unpack your nucleotides. These are your nucleotides. What are your nucleotides made of? Pay attention. So you're going to have 4 nucleotides. You're going to have A, U, C, and G. Some of them will still be attached. Some of them might be cut apart. So go ahead and cut them apart now. And then what we're going to do is we're going to match our RNA nucleotides, which are the brightly colored ones, to our strand of DNA. So we're going to build a strand of messenger RNA by using our DNA as a template. I've cut my RNA nucleotides apart, and what I have in my envelope are G's, guanines, C's, cytosines, U's for uracil, and A's for adenine. Which nucleotide is missing, and why is it missing? On my DNA strand, I see nucleotides that are bonded together, C's, T's, A's, and G's. So now what I'm going to do is I'm going to pair my RNA nucleotides with my DNA strand, and I'm going to build my messenger RNA molecule. Note that on number 3 in your lab packet, it says that you're to build your messenger RNA molecule 1 base at a time from the gene by transcribing your DNA template. Don't forget to only start transcribing downstream from the TATA box. The TATA box promoter sequence is just a TA, and that's where our enzyme, RNA polymerase, will bind downstream from. So if I look here on my DNA strand, I've got a TA. That's the TATA box. So you don't want to start your messenger RNA strand here. You want to start it after the ta-ta box. So you'll want to start it here. Please make note that this is a special nucleotide. It has 3 phosphates. Our nucleotides over here only have 1 phosphate. Keep this separate and use this one later. This is called the guanine cap. So we're just going to go ahead and put that one in the envelope and save it for later so we don't accidentally use it as one of our RNA nucleotides. As I'm building my messenger RNA strand, I want to bond my RNA nucleotides together, but I don't want to bond them to my DNA strand because this messenger RNA is actually going to leave the nucleus and go out into the cytoplasm. If we bond it via tape to our DNA strand, it won't be able to leave the nucleus. So here is my new strand of messenger RNA being built. Note, I paired, move it down. I started downstream from the ta-ta box. I paired my G with my C, A with T, C with G, A with U, and so on and so forth. And I've got a good start on my messenger RNA strand. When you're finished building your messenger RNA strand, number 4, you've just made your primary transcript. It must be processed so it successfully travels to the ribosome in the cytoplasm. So it has to leave the nucleus and go into the cytoplasm. In order for it to do that, there are things that are called intron and exon splicing, which we're not doing in lab, and we're not really going to talk about it in class. It's beyond the scope of the class, so we're not going to worry about that. You do need to add a 5' cap, which is the special nucleotide that we put inside of our envelope. And you'll also have to build a 3' poly A tail. So the 5' cap goes on the front of your messenger RNA, and the poly A tail will go way down here when you complete your messenger RNA strand. Your poly A tail is made out of a series of adenine nucleotides. So I gave you some extra adenine nucleotides. And what they're going to do is they're going to protect your RNA from degradation. So it's going to save it from breaking it down. Although poly A tails may be 20 to 100 bases long, actually we're only going to use about, I think I gave you either 5 or 6 or something. I just threw a handful in there. The next part, it asks for you to build a ribosome. Don't worry about that. You don't need a ribosome. You're going to go ahead and start filling in your packet on the next page. So the first thing you're going to do is record your gene sequence downstream from the ta-ta box of your DNA strand. So there's our TATA. We're going to go downstream and we're going to start recording our DNA strand in those numbered boxes there. So the first one would be C, the second one T, the third one G, the fourth one A, and so on. Then you're going to record the sequence of your messenger RNA strand from the messenger RNA strand that you built. Remember that you're going to go downstream from the ta-ta box. So it would be, you would enter G in 1, A in 2, C in 3, U in 4, and so on and so forth. Okay? Then it says divide the messenger RNA sequence into codons. So in order to divide them into codons, remember codons are sets of 3 letters that we read our messenger RNA, and those codons code for amino acids. The place where you're going to determine your amino acid is by your codon table. Here is your codon table. So our first sequence would be GAC. So our triplet is GAC. But we don't want to start here. What we want to do, remember all messenger RNA starts with our first AUG. So we want to find our first AUG. By golly, there it is. I went far enough that I see my first AUG. So we're going to I'm going to go ahead and pause the video. I'm going to go ahead and cut my messenger RNA right before my AUG so that I know that I don't want to use these. I'm going to leave these off, and I'm going to go ahead and start translation from messenger RNA to protein or from messenger RNA to amino acid by cutting this off.